And we will post the recorded talks on the VAP website for you to review later on, or if you want to share it with your friends and family, that will be there for you. And if you have questions, you can, at the end of the talk, either raise your hand or post your questions in the chat box. So at the end of the talk, I'll look through the chat box to see if there are any questions. Or you can also raise your hand, unmute yourself, and ask the question. So there are 10 talks this morning, and we'll start with YV Nguyen, who is working with Professor Leslie Rose. Why? Go ahead.
Hi everyone. Hey, my name is V. So today I'm gonna talk about structure function analysis of the role of lead-99 protein in asymmetric division of the C. elegans embryos. So what is the asymmetric division? Asymmetric divisions produce cells diversity, and also is a process where daughter cells inherit different components, like the color of the purple and green you see here, which leads to different developmental pathway or different cells identities A and B. This is a very important process for generating different type of cells during the normal development. And the labs use this roundworm as a model organism to study about this process because the embryos inhibit a fixed pattern of asymmetric and symmetric divisions. These are the diagram of the early embryos where different color represent different cell fate because they divide asymmetrically. The very first division of the fertilized eggs, P0 over here, is asymmetric, and then these cells divide asymmetrically and so on, and they always divide the same way. And for these asymmetric divisions to occur, a cell polarity must be established, like a point right here. And where the green cell fate is more on the one side, and then the division plan must be oriented. So only one daughter cells have the green protein. Green cell fate molecule, and the division plan is determined via correct mitotic spindle positioning. And because polarity proteins localize lead-99 to regulate spindle movement, as we see here in the wild type, lead-99 in purple localized asymmetrically on the membrane. And where lead-99 is, there's no generation force protein at that location, and the force generating complex is shown by the green color. You see right here, there's no of that, and this leads to the higher level of force generation complex on the one side, and the blood side is shown the force. This is the end result of the normal spindle orientation to have asymmetric division. And the polarity protein are conserved protein at opposite side of the cells. In C. elegans and in other organisms, they regulate the distribution of the cell fate molecules and also determine the spindle positioning. On the other hand, lead-99 mutant is the loss of function mutant with there's no lead-99 proteins, so the force generation complex distribute evenly on the cell membrane, create an abnormal spindle movement where there's no directionally in pulling force to make the rotation move, and also fails rotation in the second asymmetric divisions. So lead-99 is a very important protein for asymmetric divisions and movement of spindle. So my objective is determine which domain of lead-99 are required for these asymmetric localizations, spindle positioning, and viability of C. elegans embryos. So lead-99 is a protein that have 698 amino acid longs containing a DAP domain and a rho gap-like domain. And so our approach is to use altered version of lead-99 to rescue the mutant. So how do we test the altered version of lead-99 for function? Lead-99 is a maternal effect lethal genes. One with homozygous for recessive mutant are alive, but they all produce the all dead embryos. And lead-99 mutant must be maintained using heterozygous. We add the special chromosomes that have the pea green fluorescent protein to differentiate it with the homozygous recessive mutant when picking, and we picked heterozygous when we need to propagate the strand. Mom also created the homozygous of lead99 mutant that are not show up have the green color by like the others, and we use this to analyze the phenotype and the lethality of the embryos. So here's the different type of transgene that we use in the lab. These are different transgene were created by crossing the transgene in the lead99 mutant background. So all of the transgene that have MK of the resin protein to follow localization of the transgene to see if it can get on the membrane or not in order to do its function. So the first one have only MK at the end and no lead99. The second one is the full length of lead99 with the MK. The third one is the delta 100 to 300, which means the loss of the first 300 amino acids. And the last one is the delta 130 to 330 is the loss of the smaller region. So the transgene we're adding to the mom and then it segregate into the next generation. After segregation, we can pick the homozygous recessive mutant to do the hatch cow to see if the transgene can rescue the lethality of the lead99 mutant embryos or no. So after we do the hatch count for the live viability test, here's the result of the percent viable embryos. So the wild type shows 99.9% of viable embryos, both no lead99 and delta 1 to 300 show 0% of viable embryos. So full-length lead99 protein has 99%, while delta 130 to 330 shows 82.5% of viable embryos. And this is the summary of my result, along with the work of Laurel coach about spindle positioning and getting on the membrane. So with the no lead99, we see that has 100% lethal and did not rescue the spindle positioning. For the full length, lead99 is rescue the lethality. It can get on a membrane and rescue the spindle positioning. For delta 1 to 300, it did not rescue the lethality. It can get on a membrane, but did not rescue the spindle positioning. For the delta 130 to 330, is partially rescued lethality. It can get on membrane and can rescue the spindle positioning. So after comparing my finding with Laurel, we say that the DEP domain in the red box right here is not required to get on a membrane. However, is my necessary for spindle positioning and affect lethality of the embryos. Further experiment need to be done, and that's a part of my future direction. So I want to test the lethality of this transgenic strain from delta 8 to 110. And also I want to film the embryos from this transgenic strain at 1 cells and 2 cells state in order to observe the spindle positioning and cytokinesis. And here's my acknowledgement from my lab. And that is the end of my presentation. Thank you, everyone.
Thank you, V. Are there any questions? I have a very basic question. In your early slide, you talked about a force complex.
Yes.
Could you tell me a little bit more about that? What is the role of this force complex in the division process you are talking about, and then eventually you're testing for the lethality? So you had it in green, but I didn't quite understand what the role of that is.
I can.
Yeah,
So you say this picture. So
yeah.
the green one is where the force generating complex is at, and the first one is the blood size generate the force that is there. So it's like a motor protein that have the dynein that's pull the microtubule to the one side. So the other side receive less force. The one side
Oh,
have more force, so it can pull it over to create
okay.
a rotation move.
So this actually is referring to a physical force.
Yes, I
Uh,
have the picture I can show because I didn't show it in the right here, but I can show it right now.
Okay.
It's right here. So this is the bigger picture of the force generating complex. So
Okay,
it's the microtubule and the dynein motor that pulling in one side is, so one side have more force and the other side have less. So it can have the rotation move like this.
do you know if anybody has actually measured the magnitude of this force? Has it actually been in a more of a physical sense? You know, when they
Um, Currently we do like a scoring, we do the scoring in AV and we scoring like what is the angle that is can rotate?
okay,
Yeah,
okay,
compared to the polarity axis.
okay. Great. Thank you so much. It's such an interesting talk. Thank
Thank
you. All right, so we'll move on to our next presenter, Omnia. Can you go ahead and share your screen, please?
Yeah.
So Omnia is working with Professor Aldrin Gomes, and she'll be sharing her research.
All right. So hello, my name is Umni Ali, and for my presentation, I worked on the efficiency of combination blocking in the Western blot experiment. So just a little bit of background. The Western blot is a really powerful technique that we use to separate and identify proteins. And through the Western blot, we can also identify like the relative concentration of proteins in a specific sample. And we do that by using an antibody treatment, which will specifically bind to the protein that we're interested in. So the general procedure that we typically use in our lab, for this experiment, we use a sample of liver from 4 different mice that were 5-month female mice. And then we'll run those in a gel electrophoresis. We'll transfer the gel to a protein, the proteins from the gel to a membrane, and then we'll stain that membrane using Ponto dye. The Ponto dyed membrane will then image, which we'll later use for our analysis. And then we'll cut that membrane and block it using different blocker solutions. So this is the step that I'm going to be changing in the experiment. We'll then treat the membrane with the antibody treatment, which is incubated in a diluted buffer, blocker buffer. So we'll do that overnight. We'll wash with 1/3 TBST. Then we treat with a secondary for 1 hour, and we'll image, collect our data, and analyze. So why blocking is important? Essentially, blocking the blocking solution contains proteins, and those proteins will bind to the empty space on the membrane, so aside from our target protein. And that just allows the for primary antibody to specifically target the protein and not bind to anywhere else on the membrane. So on the bottom right corner, you'll see an example of a bad Western blot and a good Western blot. And the difference between these two is the blocking step. So the left, it's very hard to look at and probably hard to analyze as well because you have so much background noise. And so that's because the blocking solution either wasn't used or wasn't very very good. So it didn't really block. And then on the right, you'll see the Western that was blocked. So you see these very nice bands, and those correspond to our proteins. And so typically we'll use non-fat dried milk just because it has a wide variety of proteins in it and a high protein concentration. So my objective of this project was to increase blocking specificity of the Western blot and just make it and reduce background, thereby making it easier to detect the presence and relative concentrations of specific proteins. So the questions that I kept in mind while I was researching my project was first, what do we look for in a blocking solution in terms of the results? And second, what makes a good blocking solution? So what we're looking for in the results of our Western from the blocking solution is that we're blocking out all signals that do not correspond to our protein of interest. And we're doing that with low background on the membrane. And then we're also not overcompeting with our primary and secondary antibody treatments. And then what makes a good blocking solution? So typically we're looking for a buffer with a high protein concentration and a wide variety of proteins. So my hypothesis was that having a more diverse mixture of blocking solutions will better block nonspecific signals on our membrane. And so how I did this initially, my approach was to make these treatments switch. So my first treatment was a 3% milk as a control. My second treatment was 3% BSA, and my third treatment was 3% casein. And then treatments 4 through 7 are what I'm calling combination blockers. So the first one is 1.5% milk with 1.5% BSA. Second is 1.5% milk with 1.5% casein. Third, 1.5% milk with 1.5, or sorry, 1.5% BSA with 1.5% casein. And third, last is a mixture of all three. So 1% milk with 1% with 1% with 1% BSA and 1% casein. So overall, I kept a net blocking solution of 3%, and that's weight by volume. And then all of these treatments were made in 1/3 TBST buffer. So these were my buffers when I made them. And as you may have noticed, some of them actually did not fully dissolve, and those were the treatments that contained casein. So in order to try to fix this, I mixed calcium chloride at a 10 millimolar concentration and put these buffers in a water bath for a warm water bath for 30 minutes. And that helped the casein dissolve at lower concentrations, but there were still some treatments that I ultimately could not use. And so these were my final treatments. And then my results. So this is just kind of a summary of my 4 trials. Overall, there was a lot of variation. So here I have ranked for each trial 1 through 5, 1 being the most intense bands and 5 being the least intense bands. And so as you can see, there was no one treatment that consistently produced the highest intensity bands, which is what we're looking for. And then I also denoted the amount of background using asterisks. So something to note here is that BSA had a lot of background for 3 out of 4 trials. So despite having the highest intensity bands, in some cases it had very high background, which means that it wasn't really good as a blocker, or it wasn't really, it's not really easy to use for the analysis. So overall, from this initial experiment, I couldn't really draw any conclusions due to high variation. And so some possible sources of error was the casein falling out of solution. So while it initially appeared that the casein was dissolved in the treatments that I ended up using after the calcium chloride treatment, there was kind of low visibility due to the other blockers that were dissolved in the TBST, such as the milk powder or the BSA. So that kind of made it hard to see whether it dissolved. And then the casein also fell out of solution later, like after some time passed or after it cooled down in the fridge. So that's something that I considered when I was making my revised procedure. So for the second half of my experiment, I changed my treatments. Ultimately, I took casein out of the picture entirely, and my control was the same, the 3% milk for the blocking. And then for the incubation, the 1% milk, which that's how we typically do it. And then for the second treatment, I did 1% BSA with 1% milk for the blocking. And then I incubated it with 1% BSA. And that's like when I'm doing the primary and secondary steps. And then my last treatment was the 3% milk for the blocking, and then the 1% BSA for the incubation step. So my results. So for my first trial, all of the treatments, both of the treatments were significant, and they both produced higher intensity bands. So that's great. As you can see on the right, though, for the Western blot, the second treatment, which was blocked initially with the 3% milk and then incubated with 1% BSA, there's a lot of background here. So that's not too great. The first treatment, though, is looking really good. So you see these really clear bands and low background. And then in my second trial, the data was not significantly different, like statistic-wise, but you could still see like a similar trend into treatment 1, where treatment 1 performed better. Treatment 2 here, though, performed worse, so it had lower relative intensity bands than the control. So similar in the third trial, again, I had treatment 1 had the same trend where it performed better than the control. Treatment 2 here also performed better to the control, so that's more similar to the first trial. And then in my last trial, treatment 1 again performed better, and treatment 2 performed worse than the control. And this is just the trend. This data was also not statistically significant. So here's just a table to summarize the 4 trials. So only the first trial again had statistically significant data, but there's still a trend that we can see with trials 2 through 4. So overall, we see that treatment 2, which was the 1% BSA with 1% milk for the blocker, and then the 1% BSA for the incubation, very consistently performed better than the control. And then treatment 3 performed similarly to the control, but there were instances where there was high background there. So, and then to conclude, overall, the treatment 2, which was the 1% BSA with 1% milk for the blocking and incubating in 1% BSA, worked better. Some other things that you might want to consider though when you're doing Western blots in your own lab is the cost efficiency and the time efficiency. So the milk powder is cheaper than the BSA, so that's a plus, and it works pretty well on its own. And then in terms of time efficiency, it takes longer to make the like combination blocking solutions. So if you're doing a lot of Western blots in your lab, then it may not be worth going through the effort to make those solutions over and over again. So yeah. And then just a few acknowledgements. Thank you very much to Professor Gomes and our great graduate student, Gabriela Rivera. It's been wonderful working with these people. So yeah, thank you.
Thank you, Amnia. Are there any questions? So I have one quick question. So what would you say is the main reason your casein didn't work? Because I kind of, I thought casein might have worked better than BSA.
Right. So it's actually like just it's known that casein doesn't dissolve in TBSC or water. So that's why it didn't work, just because if it's not dissolved in the solution, then it won't really, you can't really use it for the blocking. Yeah.
But
it's interesting though, because there are a lot of papers with Western blots where people will use casein as a blocker. So I'm not quite sure what they're doing there, or whether they're doing using like a different kind of casein that is dissolving, or they're treating it somehow to make it dissolve. But yeah.
Okay. Thank you so much. To keep things on time, let's move on to our next speaker, Nejan Atwal. Go ahead and share your screen, please, and working with Mark Husing.
Hello, everyone. So my name is Harnesian, and I work in Housing's lab. This presentation focuses on the effects of chronic kidney disease on red islet composition. For the background, in Housing lab, our lab focuses on the islets of Langerhans in the pancreas. There's 3 main cells of the islets that we study, the first one being the beta cells. Beta cells produce insulin, and insulin works to lower blood glucose levels by promoting glucose uptake and also storing that glucose in tissue cells. The second cells that we study are the alpha cells. Alpha cells produce glucagon, and glucagon, on the other hand, works to raise blood glucose levels by promoting the breakdown of glycogen to glucose. The third cells are the delta cells, and delta cells produce somatostatin. Somatostatin inhibits both alpha and beta cells, thus inhibiting glucagon and insulin to maintain glucose homeostasis. Our goal is to study and understand how the islets in a healthy individual maintain these tightly controlled blood glucose levels, so we can understand and we can work towards finding ways to restore this control in individuals who are suffering from diabetes. For the purpose of this project, I focused on chronic kidney disease. Chronic kidney disease is a condition when the kidneys have become damaged over time, resulting in reduced efficiency in filtering the blood. The main detrimental effect of chronic kidney disease is the buildup of the uremic toxins, and these uremic toxins affect many organs in our body, including the pancreas. Based on what I have read in the literature, the 3 main effects of chronic kidney disease on the pancreas are pancreatitis, insulin resistance, and decreased insulin secretion. How it caused pancreatitis is the inflammation of pancreas, and how this happens is that one of the functions of kidneys are to excrete the hormones and the waste that is not needed in the body, but because there is a reduced efficiency of filtering blood due to chronic kidney disease, the kidneys are not able to filter those hormones and other things that it should be excreting. Thus, some of these hormones are gastrointestinal hormones like CCK and secretin. CCK specifically works to stimulate the pancreas to produce pancreatic enzymes. And if it's not being secreted, it's being built up in the blood. Thus, it's stimulating the pancreas and stressing the pancreas, thus causing inflammation. For insulin resistance, this renal insufficiency, it results in interference with the intracellular signaling pathway that's initiated by insulin. Thus, it inhibits the pathway that's causing insulin resistance. For decreased insulin secretion, these uremic compounds, they inhibit glycolysis by impacting the enzymes such as phosphofructose kinase 1, and they increase the reactive oxygen species in beta cells. Does these metabolic stressors, like what's happening in this chronic kidney disease condition, have the effect to alter the islet cell composition? And this has been shown in many altered metabolic states, such as diabetes. Thus, based on that, I hypothesized that rats with CKD have altered islet morphology and beta cell composition. So the methods that we used to get the results were, first, we prepped the rat pancreas and generated these slides. To do so, we collected the rat pancreas from Bar Lab, and chronic kidney disease was induced with an adenine-rich diet. We fixed the pancreas in 4% PFA for 5 hours. They were protected in sucrose for overnight, and then they were embedded in OCT, followed by cryosectioning the pancreas at the thickness of 14 microns. The next step was immunostaining, image generation, and analysis. For day 1, we incubated these slides with primary antibody, and we stained for insulin, glucagon, and somatostatin, thus stained for alpha, beta, and delta cells. For day 2, we incubated these slides with secondary antibody. The secondary antibody was conjugated to a fluorophore, and the fluorophore has a specific color for whichever animal's primary antibody it's targeted against. In this case, insulin is red, glucagon is far red, and somatostatin is green. Then we generated images using confocal microscopy at 60x magnification, and then we used a pipeline that was generated by two of our a graduate student and our lab manager, Mo and Naomi, to analyze the data and to quantify the beta cell composition and the diameter of the islets. Here are the results that we got. We used 5 rats for each group, 5 rats for control, and 5 rats for CKD, and 5 islets were picked randomly for each rat. The results here, the graphs shows what's plotted here are the mean % beta cells and mean islet diameter. Based on the results, we did not see a significant difference in % beta cell between the rats with CKD and the control rats. And same goes for islet diameter. We did not see a significant difference between the rats with chronic kidney disease and the control rats. This leads us to our future direction of investigating whether the reduced insulin secretion in beta cells is due to beta cell dysfunction and the effects of chronic kidney disease on beta cell health. And to do so, we will be quantifying differences in urocorn-3 staining between control versus CKD rat samples, because urocorn-3 is a maturity marker and it indicates beta cell health. Thus, a reduced activity of urocorn-3 will indicate the effects of CKD on the beta cells. I would like to thank you, Dr. Housing Lab. A big thank you to Catherine, Bar Lab, all the graduate students, undergrads, and visiting students of Housing Lab. And thank you. Sorry, I think you're muted.
Thank you. Sorry. Are there any questions? So I have a very fundamental question. You said you induce CKD, right?
Mm-hmm.
So the question is, is it possible that the induction of the CKD was not as effective as you thought? Are there other reagents you could have used because you didn't see your hypothesis didn't bear out?
Yes.
So I'm asking the more fundamental question of, did the process work? How do we know that? I mean, what made you think that the rats actually have the CKD?
So the rats were collected from a different lab, our lab, and we were told that the CKD was induced using adenine-rich diet,
Yeah.
and the rats, they did have chronic kidney disease.
Okay. All right.
Yes.
Thank you. So let's move on to our next presenter. Mazo, are you ready to share your screen?
Yes. Give me-
So Mazo also is working with Aldrin Gomes, and she'll be sharing her research.
She's muted.
Oh,
Thank
Mazo?
you. Thank you for letting me know.
Yeah, maybe you
Okay.
should.
I'll go ahead and present again. Hello, my name is Maisel. I am a fifth year majoring in neurobiology, physiology, and behavior. And today I'm going to talk about advancing Western blotting techniques. And my experiment specifically focus on adjustment of pH in twist buffer. So for an introduction, what is Western blot? Western blot is a powerful laboratory technique that allows us to detect protein from a mixture gathered from a cell or tissue, and this gives us this allows us to accurately quantify protein levels and characterize protein through the use of antibody. For my objectives, the effect of pH in TBST buffer on Western blotting, and we try to identify the optimal pH for the best Western blotting results. So for my hypothesis, the pH of twist buffer saline between 20, which is TBST, affects Western blotting. So I run the same methods as Omnia. I did run the gel electrophoresis, did a transfer blot, and then I go ahead and prepare 200 ml of 1/3 TBST, and I just dosed buffer in 7.5, 7.2, 7.6, and 8 pH, and 8. I then go ahead and prepare 3% non-fat milk and 1% non-fat milk using TBST in different pHs. I then go ahead and did the primary and secondary antibody incubation as well as washing. And then I then go ahead and do the detection and imaging using ECL or enhanced chemiluminescence. So initially I run a trial, but due to a lot of background and a lot of bench showing, I was not, it's non-reproducible, but I still wanted to go ahead and present it as it is still a relevant result from my recent trials. So for here, I cut the membrane at 37 kilodalton, and then I use a antibody proteasome subunit alpha 1 to detect a specific protein found in 25 kD. So as you can see here, there's a lot of background and bands, but we can see that bottom membrane 3 and 4 treated with the control, which is 7.6, have consistent stronger intensity bands compared to pH 8 and pH 7.2. You can see that bottom membrane 1 treated with pH 8 is very faint, as well as the bottom membrane 5 treated with 7.2. I then go ahead and run a trial, and you can see here in the membrane, due to error loading of the sample, I was able to end up having just one membrane for 7.6. And you can still see that there's a lot of background and a lot of bands, but 7.6 still show better consistency when it comes to showing band intensity and the background. So I still went ahead and do the quantification. The p-values came out high to conclude that there's a difference between the control and the treatments. But just visualizing or looking at the graphs tells us relative intensity of the bands between the control and pH 7.2 and pH 8. So when we say relative intensity, we are looking at which pH shows a stronger or darker bands, meaning that there's a higher protein concentration in that, and that means that we have better binding of antibody. Even though the results are not significant, both trials shows a downward trend. After running this previous trial, because it is non-reproducible, I run another Western blot and did my first trial using the same sample, which is a 5-month female mice, and I used liver for that. And then I treated it with the same antibody, but this time I was able to use the top membrane and the bottom membrane. And I treated the bottom membrane with PSMA1 and the top with RPT6. And these antibodies are both proteasomes. So for the first trial, you can see that I have better results, better background, and we can see that at 7.6, it's still very consistent with the strength of intensity band. And this time, pH 8 is actually showing better results than 7.2. 7.2, you can see that it's very faint comparing trial 1 and trial 2. And there's a lot of background in bottom membrane 2 for trial 1. So this shows that in different pHs, antibodies, the binding of antibody works or perform differently. And then I went ahead and do the trial 3 and 4, which is now the top membranes. I use RPT6 for this one, and we expect to see our protein of interest at 46 kilodalton. And again, I treated this with the same buffer in different pHs, and we see that pH 7.6 have very consistent intensity bands. And even in trial 4, if we change the contrast, you can see the differences in the binding of antibodies and the protein concentration. So I was not able to do quantification in this trial due to lack of time in doing so, but comparing it to the initial trials that I did, it is very consistent that pH 7 is better buffer to use when it comes to looking at primary antibody binding and background. So possible reasons for the results is the effect of pH in blocking and the effect of pH in binding antibody. So I use non-fat milk, which is also used to improve the background. We do blocking as these proteins allows to bind non-specifically onto the spaces in the membrane, and that allows the primary antibody to specifically bind to the protein of interest. And antibody is also a protein, so changes in pH could affect how it performs in basically binding to the protein of interest. So this conclusion was drawn to the very first set of trials that I did. Control 7.6 shows stronger intensity band and better background compared to pH 8 and 7.2. So the results suggest that pH 7.6 is a better choice of pH for tris buffer saline with tween 20. So thank you for listening. Special thanks to Dr. Gomes and Gabriella.
Thank you, Maisel. Other questions? So I have one quick follow-up question. So in your case, you're only using non-fat milk. You're not using like Omnia, BSA, or KC.
No, just the standard non-fat milk.
Okay. And then the other thing I noticed was between your two proteins, the 25 kilodalton and the 46 kilodalton, it seemed like you're getting a better results from for the higher molecular weight. Is it my imagination or is that an actual? It seemed like across your pH range, maybe it's an antibody difference, like you're pointing out. Maybe the antibody
Yeah,
for the 25 kilodalton protein wasn't.
I would,
Yeah.
yes. Sorry. So I would say it is the difference between the antibody that we use. When running this experiment, we assume that antibodies works differently in different pH environment. As we know that antibodies are proteins, so changes in pH could change their performance in binding into the specific proteins, as pH could change the structure of the antibody.
Great. Thank you. Thanks again to Maisel. Let's move on to our next speaker, Gurjot Gill, who's working with Bruce Draper. Gurjot, can you share your screen, please?
Hello, good morning, everyone. My name is Gurjot Gill, and I am a junior year undergraduate. I'm a junior year undergraduate in the major neurobiology, physiology, and behavior and public health minor here at UC Davis. I am doing undergraduate research at Draper Lab in the Department of Molecular and Cellular Biology. The aim of my project is to determine the role of LHX9 transcription factor in zebrafish pre-follicle cells. Now, why do we use zebrafish as a model organism to study genetics? Zebrafishes are asynchronous breeders capable of spawning all year round. Adult female ovaries contain germ cells at all stages of development, from pre-meiotic germline stem cells to mature eggs. Whereas in adult mammals, oocytes rest at prophase stage of meiosis 1, and also zebrafishes are modern molecular genetic tools for determining gene function. Here is some background information on the prefollicle cells of zebrafish ovary. Prefollicle cells surround the oogonial stem cells and oocyte progenitor cell, and then these prefollicle cells develop into follicle cells that surround the oocytes. Follicle cells play important roles in regulating oocyte development. For example, one of the main roles of follicle cell is the primary production of estrogen hormone. Now, the question arises that since prefollicle cells surround oogonial stem cells and oocyte progenitor cells, do they have a role in their development? In order to answer that question, we studied the genes that are expressed in the prefollicle cells. Here is single-cell RNA sequencing procedure in 40 days post-fertilization zebrafish ovary. Over here, we see different cell populations at different stages of follicular cell development. This is the pre-follicle cell analysis. This is stage 1B of follicle, and this is stage 2 of follicle in single-cell RNA sequencing. In this figure, we see that each dot represents one cell, and it's representing the gene expression. So similar color means the same gene expression, that this population has the similar gene expression, so does this, and so on. So over here, we see that there's these genes that are expressed in prefollicle cells, and they're progressing. And in this figure is showing the different cells that express gene GSDF. The purple color shows the expression of this gene in a particular cell. And we also see that GSDF gene is expressed in both prefollicle and follicle cells, whereas the LHX9 is specifically expressed in prefollicle cells, and that too in subclusters. We see that as soon as it's developing from prefollicle to follicle cells, the expression of LHX9 is diminishing. So the aim of my project is to determine if LHX9 plays a role in the development of these prefollicle cells. in order to study that, we did RNA in situ hybridization in 40 days post-fertilization zebrafish ovary. Over here, the RNA in situ hybridization this procedure helps us to locate the location of various cells in a given tissue and also the expression of genes in those cells. So over here, we see that the genes that are colored are being expressed. So nanos2 is a green colored. So nanos2 is expressed over here in germline stem cells, and DMC1 is expressed in the oocyte progenitor cell. And then the gray dots are the expression of DNA in zebrafish somatic cells. Over here in this figure, we see that LHX9 is being expressed in tracts of the ovary on the surface of the ovary rather than surrounding the oocytes. So these are the oocytes, and there's follicle cells that surround the oocytes. And we see that LHX9 is not being expressed over here, but it's expressed in the pre-follicle cells that are lining the ovary. So the question is, when do LHX9 expressing cells first appear in the ovary? So we tried LHX9 expression in 25 days post-fertilization ovary with RNA in situ hybridization. And in order to do that, we made a DNA template for LHX9 RNA in situ probe synthesis using ovary cDNA. So this image is just a magnification of this image above. Over here, we see that the purple color is the LHX9 gene expression, and these are the oocytes. So the follicle cells that are surrounding the oocytes, LHX9 is not expressed in those. Rather, it's expressed in the pre-follicle cells that are surrounding the oocyte, stem cells, and progenitor cells. And we see that it's only expressed on the surface of the ovary in tracts. So we did conclude that LHX9 is being expressed in 25 days post-fertilization ovary. So what is the role of LHX9 in pre-follicle cells? In order to study that, we induced loss-of-function mutations using CRISPR-Cas9 genome editing tool. And over here, we see this figure shows LHX9 DNA. And this is exon 3 and exon 4. So CRISPR-AB targets sequence of exon 4, and CRISPR-AC is expected to target the sequence of exon 4. And this is the protein. The protein is obtained after transcription of the LHX9 gene. So if there is a mutation in the CRISPR-AB, a double-stranded DNA break over here would result in mutation in the limb binding domain of the CRISPR-AB gene, which means that the protein is going to function normally up until this point, and then there's going to be a mutation that's also going to affect homeoworks. And then the other sequence that we used, like additional CRISPR genome editing, is CRISPR-AC. So if we were to use CRISPR-AC, it's targeting the exon 4 of LHX9 DNA. And for the protein, it's upstream of the homeobox. So that means if there's a double-stranded DNA break mutation, the protein is going to work fine up until this point, and it's going to induce mutation next to the CRISPR-AC, which is homeobox binding domain. So mutation was induced upstream of the homeobox DNA binding domain, and we proceeded with the CRISPR-AC procedure, and we analyzed using high-resolution melt analysis. So this procedure of high-resolution melt analysis helps us to detect mutations in the DNA. Over here, we see that the zebrafish male fishes that got the CRISPR-AC injection, they had mutations. So the red color is the wild type fishes that did not have the CRISPR injection, whereas the other male fishes are the ones that had the CRISPR injection. And we see that they have a deviation from the wild type curve, which means that these male fishes have some sort of mutation using CRISPR in their LHX9 DNA. We identified founder males with LHX9 mutations, which are these ones. And then the next procedure, here's the future directions. We have identified LHX9 as specific marker of pre-follicle cells in zebrafish ovary, and we have used CRISPR-Cas genome editing to induce indel mutations, which can be both insertions and deletions. We have identified several zebrafish founder animals that have germline mutations, and we're in the process of determining their sequence. And once we have determined their sequence, we're going to use these homozygous mutants to determine the phenotype of their progeny. Here's the acknowledgements. I thank a lot to the Draper Lab and working directly with my PI, Dr. Draper, and also AvenueB program for all their support and funding for my research project. And then for the research lab, there's funding from NIH and NSF. And you can take any questions. Sorry, you're muted, Dr.
Thank
Subbu.
you. Thank you, Gujot. Are there questions? I have a quick question. So regarding your CRISPR mutations, have you tried comparison of the previous, for example, your CRISPR AB or the CRISPR AC? I know you're targeting the home box region, but just as a comparison, would CRISPR AB
Yeah.
have shown any kind of effect? The kind of study, the melting studies that you have done to show that you're able to target the LXX gene.
Yeah, so we did both. We tried both CRISPR AB and AC. We did injections and then we analyzed the mutations by running them on the gel electrophoresis. And based on the results, we saw that CRISPR AC worked better in terms of the bands that showed up on the gel electrophoresis. So we proceeded with CRISPR AC. So we it could be done with one, but we just chose two as one can be a backup and to see that which one works better. And then the other can be a backup. So we saw that CRISPR AC, based on the results, it worked better for inducing mutations.
Great. Thank you so much. Very interesting work. Let's move on to our next speaker, Julian Flores. Julian, could you go ahead and share your screen, please?
Yes, I can. Okay. So, hi, my name is Julian Flores. I am a genetics and genomics major. I am going to be talking about my time in the Barlow lab. The project I'm working on is the project of the grad student at my lab. Her name is Judy Fishburne. And we are trying to identify the defining role of syntaxin in AOA2 patients. Syntaxin is a helicase that targets and resolves R loops, and AOA2 is a loss of function mutation caused neurodegenerative disease, meaning that when syntaxin loses its ability to do what it's supposed to do, you get this phenotype in humans, which is characterized here by these two pictures. In the bottom left, we have, if you look right next to the brainstem, you see this little well-developed web of brain matter. That's a healthy patient. And if you look at the right, there's the brainstem and this less developed web, which is cerebellar degeneration caused from this mutation. So what I kind of said it's a helicase and it resolves R loops, but what does that mean? Well, during transcription, RNA polymerase II comes in and it unwinds the DNA, and you have the nascent mRNA that's being formed. Sometimes when it's being formed, it sticks to its strand and it can't come undone. This leads to the mRNA making this R loop complex where the top healthy DNA strand is exposed to outside factors that can cause all sorts of damage and not good things. So what syntaxin does is it comes in, recognizes R loop, detaches the mRNA, and allows the coding, or allows the DNA in general, to retangle up how it's supposed to. Why are we using AOA2 as a model for study? Well, AOA2 has a prevalence of about 8% of ataxia, and it is the third most common non-Friedreich autosomal recessive cerebellar ataxia. A lot of big words. Basically, it's a common form of ataxia. Even though it is a rare disease, it is still common. It happens around the ages of 3 to 30. It's not terminal, but it is significantly life-altering. These people have troubles regulating their appetite, regulating their sleep schedules. They have trouble regulating their eye coordination. They have trouble moving. It's an issue. And a lot of these issues are characterized in other diseases as well. So if we can further understand the mechanisms of this disease, we might be able to help treat these other diseases as well. There are two hypotheses that I assist with in lab. There is the first hypothesis, which is the loss of syntaxin leads to neuronal cell death triggered by innate immune activation, a result of DNA damage. And there's a second hypothesis that the loss of syntaxin that leads to neuronal cell death can be triggered by ROS, which is radical oxygen species, or reactive oxygen species, sorry, which leads to DNA damage and innate immune activation. So how am I helping with these hypotheses and testing these hypotheses? First, I am running mouse cell experiments, which is I am running, we have two cell lines. We have a wild type cell line and a CTAX knockout cell line, which I plate onto a series of cover slips, and then I give them each their own individual treatments. These treatments aren't super narrowed down yet. We use ROS inducers such as hydrogen peroxide and BLP. And then we also use ROS scavengers such as sodium pyruvate and a few other ones that we're trying to narrow down. We do this because we want to see the different way that wild type cells and CTAX knockout cells react to these inductions of ROS and just in general how they survive. What I do is I run these experiments first. I've ran 3 of these individual experiments. There's many other experiments that I have not ran, but I also do a lot of our imaging and our analysis, which is poorly shown here. These are some of my earlier images, so they're not the best, but on the left you have just a regular DAPI screen where this is just showing the cells, and then on the right you have a gamma H2AX filter, which is showing particular foci of damage. Gamma H2AX is non-specific though, meaning that it's going to show DNA damage of both single-stranded breaks and double-stranded breaks. So the way we filter to actually determine which kind of break is by foci intensity, which is part of the analysis part, where me and Judy will come up with a number that we set as a threshold for the experiment that actually determines a good, like this is a double-stranded break. And we're looking for those specifically because, as I referenced earlier, when there's not good R-loop resolution, the way DNA repairs that is through snipping at both ends and then doing non-homologous end joining, which is what we're looking for specifically, that kind of DNA. ROS induction in general can just lead to other kinds of DNA damage, so we aren't trying to look at that. I also, on the side, do genotyping and cell culturing in the lab, which is we have a mouse culture or mouse center facility that we keep a bunch of mice alive in. I do a decent amount of the genotyping to make sure that these mice are what we think they are. And then I also do cell culturing where I'm passing cells and making sure they're healthy, swapping out their media and feeding them and do everything like that. And I have some pictures of that. The genotyping I do through gel electrophoresis, as was pointed out by many other people in this stuff. It's a common thing. And then that's just a picture of the incubator. I don't want to take them out and potentially contaminate the cells. So yeah, that is my time at the Barlow lab. Any questions?
Thank you, Julian. Are there questions? So the one very basic question I have is your hypothesis regarding syntaxin.
Yes.
The images you showed of the mice cells, could you please explain a little bit more about how this bears on that the syntaxin? Because I kind of lost it in this description.
Yeah.
I see the black and white cells, and then there is a fluorescent probe stain cell, I believe. So yeah.
So the two hypotheses is that one, our wild type cells have a lower basal innate immune activation, which these images don't necessarily show this because I'm not showing these compared with wild type and CTAX, which is my bad. I should have done that. But what we would do to compare the two is we would take basal levels of fluorescence on the right and compare the foci concentrations per cell. We do this for our like negative control where there's no treatments. We count 100 cells and we count how many foci per cell there are, and we compare the basal immune activation based off of that, if that makes sense.
Okay. All right.
Yeah.
Thanks, Julian.
No problem.
And let's move on to our next speaker, Francesca Dallara. Could you please share your screen? And Francesca is working with Neelima Sinha.
Okay, hi. So hi everyone, my name is Francesca Delara, and my presentation is on mutagenizing the common bean. So just a little bit about me. I'm currently a fifth-year transfer student at UC Davis who is majoring in plant biology and minoring in public health. And my hobbies and interests include in being outdoors, playing instruments, botany, and podiatry. So the lab that I am currently in is called the Sinha Lab. My PI is Dr. Nalima Sinha, and my lab manager is Vicky. And the focus of our lab is on the development of leaves. So the aim of my research is to test different methods to mutagenize or edit beans, since beans are very recalcitrant, meaning that they aren't very cooperative. And the main method of infiltration that I used for my study is smushing, which you can see on the far right corner picture. And then the beans that I worked with throughout the study were Canarios and Montcalms. So as I had previously mentioned, beans are very recalcitrant, so their transformation and the genetic editing process is very challenging due to the fact that it's a lengthy process, the efficiency is very low, and it's cultivar dependent, meaning that depending on the bean, they perform differently. And beans are also not very cooperative when it comes to in vitro methods, such as in terms of regeneration and rooting. And the significance of my study is to be able to create beans that are more resilient to both biotic and abiotic stressors, which will hopefully lead to the abundance of beans that will be able to help feed our growing population, because we'll be able to basically like pick out more desirable traits. So just some background information or some terminology that I think will be useful throughout the presentation. First off is CRISPR. It's a genetic editing tool that allows us to make changes or edits to an organism's DNA with a specific targeted DNA sequence. And there are two components to CRISPR, and that's the guide RNA and a Cas9 protein. A plasmid is a small circular DNA that's introduced and integrated into the DNA of the organism that will then partake in the plant's genetic phenotype or genotype, and it will also lead to specific desirable traits. And the PDS gene is a homologous gene that's present in all plants, and it's essentially the gene that our plasmid is trying to target and edit. And if you knock out this gene, which essentially makes plants green, if you knock it out, then you'll end up getting like albino plants, which you can see there's like white plants on the far right picture. And then lastly, the plasmid that we used is called PMR913, and it's the plasmid that contains the Cas9 protein, two guide RNAs that target the certain PDS gene, and then a GFP marker for expression. And you can see just like a visual image of our plasmid on the left picture. So some parts of the plant that we worked on in the project, so just a little bit of some plant anatomy for you guys. So starting from the top part of the plant to the bottom, we have the first true leaves, which are these big leaves. And then we have the epicaudal, the cotyledon, and then the hypocaudal, and then we have the root system. So along with our research, we used a scientific paper written by Li Yang and among with other authors. And essentially in this paper, they worked with the lab rat in like the plant world, which is called Arabidopsis. And they basically made their own transgenic plants. And with that, we used their mobile sequences to help us move our CRISPR elements from the transgenic root system and onto the shoot system of the plant. So beginning with the methods section of the study, so I first created a incision at the epicaudal of the plant using like a razor blade. And then next I would take that top half of the plant where the incision was made, and I scratched it or I scored it in order to increase the surface area and the susceptibility of the plant to the bacterial infection. And then I took that incision, I smushed it or rubbed it against the agrobacterium that contains our plasmid, PMR913, and I made sure that that incision is completely coated with that bacteria. with the plasmid. And then lastly, I just placed the plant in a new pot with vermiculite soil and allowed it to grow for roughly 2 weeks. So after allowing the plant to grow for several weeks, I then collected the plants and I observed the changes and the growth of the root system that occurred or that's present under the incision. And then I basically checked or did a quality check of GFP. And GFP is basically a green fluorescent protein that exhibits green fluorescence when it is exposed to a certain wavelength of light, and it's usually within the blue to ultraviolet range. So as you can see on this, these are the same plant. It's just that the picture on the left is the plant under like normal lighting, and then the plant right here on the right that shows the GFP expression within the roots is under a certain UV wavelength. And I just showed a jellyfish because the GFP is, or like jellyfish produces, or it's from, or creates GFP. So just because we see GFP in the plant, that doesn't entirely mean that our edits were successfully made. So we then performed a PCR on the PDS genes from the collected DNA from the plants that show GFP. And so I just want to mention that our constructs can only do certain mutations, and those are deletions or point mutations, meaning that our DNA will get smaller or shorter if the edits were made. And so as you can see in sample number 1, there are 2 bands, and you can see that the band, one of the band on the bottom has moved further down the column, meaning that that DNA has gotten smaller or shorter as compared to the wild type. So moving forward or looking into the future, we hope that our CRISPR elements will move into the shoot system of the plant, and hopefully we will then be able to observe some edits. And then we also hope to collect seeds from these plants and successfully observe edited progenies. And then lastly, we also hope that this method will be applicable for other beings and will allow us to select for desirable traits. Thank you.
Thank you, Francesca. Very, very interesting talk. So, other questions? I have one question. So tell me again why you chose the 2 genes. You chose the Cas9 and the, sorry, the other one. I forgot the name of the other gene. But if you could maybe go back to that slide and explain to me one more time as to why you're targeting those 2 genes.
Yeah, sorry, let me go back to, so there's only really one gene that we were targeting, and that's the PDS gene. Sorry, I'm like trying to get this. Okay.
So is the Cas a control then?
So the Cas9, like in CRISPR, that's basically like what the
Yeah, right. Okay, yeah, I understand. Yeah.
but the PDS gene, the reason why we're targeting that one, sorry, I'm trying to pull it up, but okay.
No, you can just tell us. Doesn't matter.
Okay. Okay, yeah, so basically the PDS gene is it's homologous, so you can see it in like most plants, and that's essentially what that's essentially the gene that makes plants green. So it's like important for like the chlorophyll, the chloroplast. And so basically if we do target that PDS gene, it's an easier way for us to basically detect if the edits were made, because if it targets the PDS gene, then we'll know that it was like edited or targeted because the plant like didn't turn green. So that's why I like showed in like one of my slides that if you knock out the PDS gene, then you'll get like albino plants, like which are like plants that were white. Sorry, I'm like having a hard time trying to pull it up, but
It's okay. All
yeah.
right. Thank you. So we'll move on to our next speaker, Jaspreet Kaur, working with Karen Bales. Jaspreet, could you share your screen, please?
So my name is Jaspreet, and I work in Dr. Bale's lab, and I'm a fourth year human biology major. And my project focused on the differences in parental care in naive and experienced prairie wolves. So I just have some background about like what the Bale's lab works on. So there's like many projects, and we work with like a bunch of different animals. So we have like titty monkeys, lion seahorses, and prairie wolves. But I work with the prairie wolves, and I look at pair bonding between the prairie wolves in healthy aging and doing breeder ranking as well. So breeder ranking is like a term that we use just in like the Bale's lab, and that's where we're ranking the behavior of parents and when they're interacting like with their pups. So we rank their behavior, and then when the foals are born, we like keep them for 20 days and then we wean them. So for my project, my hypothesis was that the older pairs will have lower contact times overall compared to like the naive new parents due to increased amount of experience with parental care in their past litters. So why did we start this project and why do we study this? So one of the main reasons was like during COVID, we had like lack of human interaction with like other individuals, but there was like a higher increase in like at home where parents were forced to spend more time with their children. So we use prairie wolves because they're monogamous animals, they're biparental, they're really small, and they're able to have a lot of litters. So on the right, I have like a picture of like a prairie wolf. So they're really small, so that's why they're also easy to use. So when we do breeder ranking, we perform this test like within 3 days that they're born. And we do this observation mainly for 2 litters, but sometimes we may have to do it for like a 3rd litter. And this test is completed through like an online computer system that's created by Dr. Bales and her colleagues. So like, for example, if pups are born like on a Tuesday, then we rank them AM and PM for the next following 2 days. We look at maternal and paternal observations like simultaneously. And for our data, we save it as like high, low, and low, high, and medium. And what this means is that like when we finish breeder ranking, we have like a total contact time, and sometimes we could put it through like a system that's gonna spit out like, is it low, is it medium, or high? And then if the first 2 litters don't match, like on the right, I kind of have like a little flow chart that kind of shows like how we like go like day 1 AM PM and day 2 AM PM. So if these first 2 don't match, then we have to rank a 3rd litter. So these are some parental behaviors that we notice. So on the left, I have female behaviors, and then on the right, here are some male behaviors. And then there are some behaviors that both male and females can do. So in regards to like the male behaviors, we have huddling and non-huddling. And huddling is when all 4 paws are touching the ground and the male is huddling over the pups. And then non-huddling is basically the opposite of huddling. So that's when they're not doing huddling at all. And then for female, we have active nursing, which is when like the female is walking around and like the pups are attached to her. And then lateral nursing, so the female is like on her side. And then neutral nursing is like when the female is on top of the pups and she's nursing them. And then in regards to like the behaviors that they both have, there's licking and grooming, and licking and grooming only refers to like the pups. So when we're licking and they're licking and grooming just the pups. And then retrieval and removal, so licking the pup at least 1 inch above the ground. And then non-pup directed behavior is anything that includes nest building, eating or drinking, or auto grooming. And then we also have a function where we observe like if they're going in the nest or out of the nest. So how far have we come? So in early April, we finished collecting all the data from my project. So we had a very limited like time span to collect everything before the undergraduate research conference, but we were able to make some graphs to use. So one of the things that I showed on during like the research conference was looking at total contact time. So this was the total contact time for like old and new parents. And then on the right, I have the graph that I use, which shows on the x-axis the experience. And then on the y, I have time spent in contact, which is the same thing as total contact time. So what did our results show? So our data suggested that total contact times between experienced and naive parents was not significantly different. So we had a p-value of 0.12. However, the data did elucidate that if we did do further investigation, we could potentially observe like a trend and maybe like a potential difference. So the next step is to still use the same like data that I already have, but maybe compare like male and female sex ratios. Like maybe we'll see a difference between the experienced parents and then the new parents. And then this is just the poster that I presented at the conference, and then the grad student, Sophia Rogers, who helped me with this project. And then I would like to thank Dr. Bales for letting me be part of her lab, and I will be continuing being in her lab even after I graduate. And then I would like to thank Jessica Bond, our lab manager, and our lab scientist, Adele, and then Sophia Rogers, who is a grad student at the Bales Lab, who's helped like me throughout this entire process, and then the VIP program in AvenueB. Thank you.
Thank you, Jasprit. Are there any questions? I have some basic questions. So what is the typical lifespan of a prairie vole, and how often do they have litters? And what is the nursing?
So I'm not quite sure like what the typical lifespan of the prairie vole is, but they do have like offsprings quite often. Like usually after we wean the pups away from the parents, we typically do see that they can have new pups within like even like couple weeks, or but it really just depends on the pair, because for some pairs we didn't have any like offspring for a long time, and then some like we see that they have multiple.
So typically the nursing period is what? Month?
Sorry.
How long do they nurse their pups for you to?
They usually nurse their pups for at least 20 days until we wean them.
Okay, so your observations are over a period of 20 days regarding
No.
the behavior.
Yeah, that's a good question. So when we're observing them, we only observe them within the first 3 days that they're born.
Ah, okay. And in those 3 days, you're saying that the difference between a naive versus, or the new versus an experienced mother is not a big difference.
Yeah, so far we've seen that there isn't a huge difference, and this may just be due to the lack of like, because we didn't really collect too much data just because we only had 10 new cages, but it did elucidate that if we did have more cages, potentially we could see a better trend.
Okay, great. Thank you, Jasprit. Very interesting data. All right, we'll move on to our next speaker, Dustin Lower. Dustin, could you share your screen, please?
I can. All right. Good morning, everybody. My name is Dustin Lower. I'm from the Boger Lab in the Department of Plant Biology. And today I would like to tell you about my projects about how wildfire affects the symbiosis between tree seedlings and soil fungi. So we all have seen the increase in wildfires in the last year, all the smoke in the air, and we typically think, okay, well, how does this impact like just the forests, which is important, especially for restoration, but we really don't know much about how it affects soil fungal communities. And I work in a mycology lab. I have been a victim of a wildfire, so I wanted to look more into how this affects the symbiotic relationship between fungi and trees. So when we think of fungi, we typically think of mushrooms. but the main body of the fungi is actually underneath the ground. It's these network of filaments individually that are called hypha or hyphae, and together they come together to create the main body that's called the mycelium. Now, the mycelium, usually it's recruited to plants using root exudates and phytohormones. So the mycelium grows towards the plant root. It actually penetrates the root cortex, and the hyphae, as you can see here in green, actually grow around the root and into the root apoplastically and create what is called a fungal sheath around the root, and then inside the root, what is called the Harding net. And the Harding net allows for nutrient transfer back and forth between the plant and the fungus. So mycorrhizal fungi, which are symbiotic plant fungi, they colonize the roots, like I said. The sun beats down the photons, the plant takes it up, photosynthesis, all that good stuff. So in return for the photosynthate that the fungi receive from the plants, the fungi will funnel things like phosphorus, water, and nitrogen into the plant. So it's a true symbiotic relationship. But a fire comes through. And when a fire comes through, it kills the trees, as we know, heats up the soil, and kills all the fungi and the plant life. So I wanted to kind of look at, well, what happens to these symbiotic relationships after a fire rolls through, and how these relationships are reborn after the fire. So what I did is collected some soil up from the Sierras. We did 4 different burn treatments, one that was a high severity fire, one that was a low severity fire that happened more recently in 2022 and 2023. There was another low severity prescribed burn that burned in 2002, 2009, and 2017. And we also collected a control in an area that hadn't been burned in at least 100 years. So I conducted a bioassay in the core greenhouse complex. You can see we use kind of like a block randomization setup where the different burn treatments were like randomized throughout the table in here. Like the different colors on these cones coincide to the different burn treatments. And as with most ecological research, you plant the seeds, you have to wait for it to grow and kind of see what happens over a period of time. And since I'm doing mycorrhizal research, we had to wait at least 9 months to really be sure that the mycorrhizae had colonized the roots. So in the meantime, we had done some soil analysis, looking into soil pH, because fungi typically prefer a little slightly acidic soil. We wanted to see how the soil pH had changed after the fire, soil mineral content, soil organic matter, Bray-2 phosphorus, which is total phosphorus in the soil, and then carbon to nitrogen ratio, as the C to N ratio for fungi is typically higher on the carbon side to the nitrogen side. So we wanted to look into that, and as well as soil texture to see if any basic soil texture had changed post-fire. Other data streams that we're looking into is the plant biomass after the bioassay grew enough to where it would have the colonization, harvested everything, separated the root from the shoot, took the dry and fresh biomass of the plants to see which treatment had more success in growing. The germination rate between the different treatments was also noted. Soil nutrients, as I mentioned before. I'm going to be measuring the tissue nutrients on the seedlings here soon. And I'm also taking care of community composition and percent colonization. Community composition will kind of tell us like what fungi are there and in what concentrations, and percent colonization essentially tells us on an entire root system what percent of those root tips are colonized. So you can see kind of here in the bottom right on the picture on the left, that would be an uncolonized root, where all those other root tips there, those are colonized. It kind of has like this bulbish structure. Sometimes it's like a fluffy white like there on the right. Sometimes it's like more like a dark color like you see on the left, and it just depends on the species of the fungi. But these are the structures that the fungi make that allow for the nutrients to transfer back and forth. And typically you want about 50% colonization is pretty average. So anything less than that, especially if it's like much less than 50% in like any of the high burn severities, will give us a pretty clear indication that the colonization of the fungi was highly impacted based on the severity of the fire. So overall, my hypothesis was that fire severity can be directly linked to percent colonization, community composition, diversity, and that the more severe the fire, the more highly impacted that the soil fungal communities would be, and then that would in turn affect long-term restoration efforts for that particular forest. So the first thing I looked at was germination rate over 55 days with number of germinates on the y-axis and days since planting on the x-axis. The different colors are just the different burn treatments, with the white being never burned, green being one of the low ones, and yellow being the low old one with the least recent fires. The red being the high severity fire. And as you can see, there was a definite impact on germination based on the severity of the fire. And that kind of gave me a little indication that I might be onto something with regards to fire severity. As I mentioned before, soil organic matter is important for fungal communities. Usually the higher your soil organic matter, the more indicative it is of a robust fungal community. As you can see, the never burn is considerably higher, the low new lagging a little behind, and then the high severity and the low old are, at least half of the other ones. And this was from the soil that we collected from the mountains. This was more just kind of seeing what was going to be indicative of the results that we get later. And then here's the carbon to nitrogen ratio that I mentioned earlier, where higher carbon to nitrogen ratio is better for soil fungal communities. It said that there's kind of like a golden ratio where it's like 21:1 carbon to nitrogen is ideal for soil fungal communities. And again, you can see there is a pretty sizable difference in the soil that had never burned compared to the other burn treatments, where I will say I did expect the high burn to be significantly lower in carbon to nitrogen, but you can see that fire alone does impact this, this ratio. Then there's Bray-2 phosphorus. Typically with Bray-2 phosphorus, the higher number you have, the more indicative of an unhealthy soil fungal community. If there's a lot of phosphorus in the soil, that means that the fungi aren't really taking in that phosphorus to funnel it to the plants. So a high Bray-2 phosphorus number would be indicative of a poor soil fungal community, whereas you can see at 12 with the never burn, those fungi are taking in that phosphorus and trading it to the plants for their photosynthetic carbohydrates. So I did take the biomass data. I still have to analyze that. I did the percent colonization. I took that data down, but as of right now, I'm up to my eyeballs in PCR and gel electrophoresis. I have 500 samples that I have to process still, get those run through gels, and then send them out for sequencing so we can test the community composition and see what's in there. It's a little time sensitive with the tissues that we have, so I wanted to get that out before I went back and started analyzing the biomass data and the percent colonization data. As I said before, I still have to process some tissue nutrient samples where, I already dried out my samples. I'm going to grind them up, kind of homogenize them, and we will send those out to a lab, and they'll be able to get back to us the data on if the tissue nutrients in these seedlings was impacted based on the soil treatment that I planted them in. And I'm also currently working on my manuscript for publication, which I hope to finish by the end of the summer. I'm going to be continuing this research through the summer. I'm going to be staying in the same lab that I'm at. I'm actually going to be also doing my PhD research in the same lab. So a lot of what I've been doing in the last year and a half will be continuing on indefinitely into grad school. And I would like to thank VIP and LAEP, AvenueB, and my fantastic PI, Dr. Laura Boger, who couldn't be here today. She's on paternity leave, which is a good reason for not being here. But she's been super supportive and has had my back every step of the way. And I couldn't ask for a better PI. And I look forward to working with her more in the future. Thank you.
Thank you, Justin, and for a very interesting talk, and congratulations on continuing your research for a PhD.
Thank you.
That's really wonderful to note that this research has led you to a PhD degree.
Yeah, the same research that I'm doing, I'm going to be doing my PhD research on. I love it.
So I have two questions. One,
Yes.
what is the germination plant or the plant that you're germinating in these various soils? Is it the pine or?
This is Douglas fir.
Douglas fir, okay.
Yes.
The one other thing that was curious for me was between the high burn soil and the medium or the low burn soil, what I noticed among that second category of soils where you had the old versus the new, it seemed like many of these things that you're measuring, whether it's the CN ratio, phosphorus, or the organic matter, it seemed like the old and the high burn seem to track each other. In other words, when I look at those graphs, the new ones seem worse off than or I can't tell why the old versus the new, the difference you are seeing.
Yeah, there wasn't really too much different between the old and the new. Some of the literature review that I had done, some of the statistics were kind of indicative of there being like a temporal aspect in the change in soil nutrients, where they would test the soil right after the fire and it would be like at one level. They would test it, a few months after that, and it would seem like it was going up. And then they would test it again after a couple of years and it went down again. It's really kind of like this roller coaster ride of biogeochemical processes. So I just kind of wanted to add that in there too, to see if I could glean anything from a temporal aspect from time since burn.
Okay, great. Yeah, that was kind of interesting to see the difference between the old and the new. But this is, I believe, the low burn. Is it the low
Yes,
burn?
yes.
Okay. Well, the high burn, I guess you never compared is it all just high burn? There's no such differentiation there, the low versus the new versus the old?
Um, I haven't found anything significant in the new versus the old. There's like this the US Geological Survey and the USDA has what they call like their burn portal, where they actually use satellites to track burn severity. So I think in future research, I'm going to try to find multiple severe fires, and those severe fires that burn, maybe like one was like super severe, like the highest temperature that you could imagine from a wildfire, and one that's rated severe but maybe not as severe as that one, and kind of compare different severe fire gradients, I guess you could say. Because I mean, it just kind of stands to reason that a severe fire is going to have more of an impact than a low intensity burn. So I think I'm going to focus more on the high intensity, because I want a lot of my research to focus on biodiversity, because in soil ecology, the more biodiversity you have, the more it impacts soil multifunctionality. So all the functions that these fungi do, they impact the entire soil ecology. So I want to see if the gradient of burns impacts biodiversity, which in turn impacts like their function in the ecosystem.
Thank you so much. It's really interesting work and it's very important work.
Thank you.
Okay, we'll move on to the last talk of the morning, Damien Murillo, who is working with David Furlow. Damien, could you please share your screen?
Give me 1 second. Let me pull it up. There we go. Okay, so my name is Damien Murillo. I am a current 4th-year biochemistry and molecular bio major, minoring in human physiology. I'm also working in the Furlow lab, where my project was focusing on the role of thyroid hormone receptor beta in the development of the allotetraploid Xenopus laevis. And the frog in the background is actually a picture from ZenBase, which is a collective of various information on Xenopus. So just wanted to include that just so you have a reference of what the frogs look like. So just a brief background on these frogs and why we're interested in them. It actually has a very deep history in biological sciences. They've been used for studies when it comes to looking at development from fertilization. I believe they were used in the discovery of the CDK mechanism for cell regulation and the cell cycle, but also they had been used as pregnancy tests in a more applicable way. I believe in the 60s or the 70s, at least before more normal pregnancy tests came around. So these guys have been kind of the forefront of a lot of different experiments that go on. And the unique thing about Xenopus is it's what's considered allotetraploid. So instead of being diploid like us, so we have 46 chromosomes, 2 copies of each chromosome. Xenopus has 4 copies of each chromosome, specifically Xenopus laevis. So evolutionarily, this goes back to the most common ancestor, which diverged with Tropicalis back 48 million years ago. So there was a divergence event between Xenopus what would become Xenopus laevis and borealis and Xenopus tropicalis. Tropicalis has 10 chromosomes and is diploid, and laevis has 9 chromosomes and is allotetraploid. So that's probably where that event occurred. And then there was some speciation event where the L Xenopus and the S Xenopus, roughly 34 million years ago, diverged. And then there was another fusion event where the L Xenopus and the S Xenopus had mated and created an allotetraploid Xenopus laevis roughly 17 million years ago. And so it's kind of interesting because it's allotetraploidy inherently means that, you know, there's not one quote unquote genome, there's actually 2 what are referred to as subgenomes. So the L and the S are what we refer to them as right now. And so if we look at this diagram, the XTR is Xenopus tropicalis and XLA is Xenopus laevis. So for example, if we look at chromosome 1, Xenopus tropicalis 1 and Xenopus laevis 1L and 1S, they're homologous chromosomes. So we use the common numbers that are used for tropicalis in order to denote which copies of chromosomes they are. And then we actually use the L and the S to specify which subgenome we're talking about. So in this case, L is literally referring to long and S is short, just based on the chromosome size. I mean, in this diagram, you can kind of see it. If we just look at the first one, the L is a little bit longer than S, chromosome 4, L, a little bit longer than S. So that's kind of the terminology that we use when we refer to genetics for Xenopus. And the reason why Xenopus has been used so much in research is because the metamorphosis from going from a tadpole into a froglet stage is a entirely thyroid hormone dependent process. So the thyroid system is highly conserved throughout all vertebrates. So anything that works in a frog, works in a fish, works in a dog, works in a human. So this is a really, really great animal to look at if we want to understand how the thyroid system plays a role in development and, you know, maintenance of life throughout the lifespan of the organism. And in addition, the metamorphic progression is very distinct. So, you know, you're going from this stage 45 free swimming tadpole where you have a tail, you don't really have any limbs, you have a very small head, and you're going to this frog. So there has to be reabsorption of the tail, there has to be generation of the limbs, a little bit of broadening of the head, growth of the entire body. And then there's also a lot of remodeling that's occurring within the actual organism, one of which is the intestine, which I will get into in a bit. And so this is why we kind of use this animal as a model organism. In addition, they're extremely hardy animals. In fact, they're considered an invasive species in California. LA has a big problem with them. And recently, I believe Golden Gate Park had an issue with them and they couldn't really exterminate them. So they just quarantined off the area and had to let them die out because they are just so good at living and invading other regions that are not their native region. In addition, they spawn a lot of eggs. So I mean, when we do our matings, I think it's anywhere from like 400 to 500 is kind of the average, at least for wild type animals. So these are very, very good models to use. So, you know, the common insight for why we're diploid organisms as opposed to being haploid is because, you know, having 2 chromosomes protects against degradation. If one of our chromosomes are messed up, the other one can kick in and we're fine. So then that kind of begs the question, why does this animal need 4 copies? What is it doing with those extra copies? And so in order to try and answer that question, my hypothesis is reducing the number of functional copies of the thyroid hormone receptor beta gene will result in reduced growth of animals compared to wild type. And so one of the main organs that I looked at was the intestine. So in this diagram on the right, if we look at the expression of thyroid hormone receptor beta in the intestine, specifically the L allele, it's much higher than the S. So it makes it a very good study for looking at specifically the L allele versus the S allele. So in order to kind of quantify this quantify the response to T3, tetraiodothyronine, which is considered the active form of thyroid hormone. We did a variety of experiments. So the 1 week assay to measure response to precocious exogenous T3. This was adapted from Mengele et al. 2018, who was actually the assistant project scientist in the lab. And in order to determine the genotype, we actually have to do PCR. In addition, we did an NF54 assay. So NF just is the common staging criteria that's used for Lavis. NF54 is right before metamorphosis begins. So they're essentially primed and ready to go. They haven't begun producing endogenous T3, at least in highly detectable levels. So this makes it a really good stage to hit them with T3 and see what happens. Can they respond and how do they respond? We can also compare post-metamorphic growth rates and pre-metamorphic growth rates. And we're also doing RT-qPCR of specific thyroid hormone response genes that play a role in metamorphosis, just to see if, knocking out different numbers of these copies of chromosomes, does it directly relate in a reduction in gene expression? Does it elevate gene expression? So this is kind of what the PCR looks like. These are probably two of my more beautiful gels. So on the left image on the top row, this is going to be the L wild type, and then on the bottom row is the L mutant. And so what that refers to is we entirely knocked out the S copies of thyroid hormone receptor beta. So prior to me coming to this lab, using TALEN modification, they were able to selectively knock out the S alloallele entirely. So we were just left with the L subgenome. And then they introduced just enough of that TALEN to knock out one of the L chromosomes, but leave the other one intact. So we amplify around the L mutant area because with the TALEN modification, it created an 8 base pair mutation where effectively they were removed, causing a frameshift mutation. So we're able to see in the genome of the animal where the mutation occurred. And that gives us the ability to see, okay, is this a wild type chromosome or is this a mutant chromosome? And so we commonly refer to these animals as either L wild type with a S null background, L het with an S allogenome knockout, and TR beta knockout, because if we knock out L with the S background already knocked out, then it's just lacking thyroid hormone receptor beta either way. And so with our 1 week assay, this is kind of what we were looking at. So one of the things that we look at is the head area. So J strain is what we considered our wild type. It's a highly inbred species or highly inbred line of these animals. So they're going to have both the L and the S alleles. This is our wild type and our DMSO treatment, which is just our vehicle control. This is the L het animals and DMSO treatment, and then our knockout animals. And so they kind of all look the same, which is what we're going for. At this stage, their development is pretty much all aligned. And then we hit them with 25 nanomolar T3 and see over a 2 or 4 day span how their development changes. So with the J strain, when they're exposed to 25 nanomolar T3, there's a great reduction in the head area. And there's also an angling of the jaw, kind of seen by that peak here. And then if we look at our L het, where they only have 1 copy of this receptor, it's compared to the L het DMSO, the head area does shrink a little bit, and then you start to have angles forming in the jaw, but not as drastic as the J strain. And then TR beta knockout, it kind of looks identical to the DMSO treatment. So that just verifies that if we don't have TR beta, T3 isn't going to do anything because it can't bind with any affinity and it can't do anything. So we did the 1 week assay using DMSO and 25 nanomolar T3, the J strain wild type, and the L het S nulls and the L nulls S nulls, which are TR beta knockouts. And so if we just look at J strain, which is the black boxes, between the DMSO and 25 nanomolar treatment, there is a great reduction in head area. And then if we look at the L het animals, between DMSO and 25 nanomolar. There's a slight reduction, but not as extreme as with J. And then if we look between DMSO and 25 nanomolar for the TR beta knockouts, they're virtually the same. I mean, there's very close overlap. And then we can compare between treatment groups as well. So with 25 nanomolar, the J is very low in hit area, but TR beta L het is much greater, and then TR beta knockout is a lot higher. In fact, it's the highest of all of them, showing that without the receptor, you can't respond to T3 very well. And so with the NF54 assay, we went ahead and measured intestinal length. And this is actually data I just finished up polishing this morning. So this is also new to Dave, my PI. So if we look at the J strain with DMSO treatment, that's kind of the standard. And then with introduction of T3, so we did 10 nanomolars of T3 over a 2-day span. There's a clear reduction in intestinal length. And so what happens during metamorphosis is the intestine has to go from the larval intestine to the adult intestine. And the larval intestine is much, much longer. It has a bunch of coils that it coils in on itself. And then over metamorphosis, it slowly condenses into the adult intestine, which is much shorter. And with the L het, we also see that going on. So you have reduction in length with the addition of T3. Now, what was really strange to me was in the knockouts where they shouldn't be able to respond to T3, they still responded to T3. So honestly, I don't think that's a failure of the animals, but I think it was a failure of techniques and methods, because this is a method, or rather the extraction of the intestines was a protocol that I kind of had to write myself and I was testing out along the way. And so I think variations in the protocol and slight changes that I did between protocols and between samples, I think that may have messed with the numbers. But I still need to do statistical analysis on this to kind of see what was going on. So next steps, right now we're doing RT-qPCR. That takes quite a bit of time between making RNA and then carrying out the qPCR. But overall, we can see that, at least according to the 1-week assay, removing thyroid hormone receptors in the L hets and the TR knockouts, it creates a blunted response to T3. So you don't have as much shrinking of the head, you don't have as much angling of the jaw, and effectively they're just not able to respond to T3 very well. And so far, the data from the NF54 assay is inconclusive. One of the things that I've noticed a lot, specifically with Yunbo Shi's paper, Yunbo Shi actually did a lot of these experiments in Xenopus tropicalis, is that he normalized them relative to snout to vent length. So they divided the intestinal length by snout to vent length, which is something that I did not do. I did not measure snout to vent length. I didn't think to do that. But in the future, if I decide to do this again, that's definitely something I would take into consideration, just so we're making sure that they're being normalized. And next steps, I would like to compare the S and the L subgenomes. So there's slight differences in the sequence for thyroid hormone receptor beta between the S and the L subgenomes. And there hasn't been a lot of research done on that, mainly because it's really, really hard to get those isotype specific changes. It's just very difficult and it's impractical right now. But this could be achievable via almost identically to what we did. So 1-week assays, monitoring growth, looking at mortality, RT-qPCR. And yeah, just looking at more of the receptor isotypes, looking at beta versus alpha, and then, beta L versus beta S, alpha L versus alpha S would be the overall goal to understanding the thyroid regulation within this animal. And so that's kind of my presentation. Special thanks to Dr. David Furlow, Dr. Brenda Mengeleg, Mika Taylor, which was an undergraduate that helped me a lot during my experiments, and AvenueB for the support throughout pretty much all of it.
Thank you, Damien. It's a very interesting talk. Are there any questions at this point? So I have one quick question on the 2 pairs of L chromosomes, right? You have 2 pairs. You knock out one. So what happens if you knock out both?
So there are 2 copies of the L chromosomes. So there's 2 pairs of the TR beta. So I believe it's on chromosome 6. So there's 4 copies of that chromosome. There's 2 S chromosomes and 2 L chromosomes. And so what we did in this experiment was actually knock out one of the L's and then both of the L's.
Right. And you also mentioned one of the future objectives of being knocking out the S as well.
Yeah, so we did everything right now was with an S null background. So we completely ignored the S allogenome. And so what I would like to do instead in the future is knock out the L genome altogether and just focus on S and see if there's any differences there compared to the genome.
Okay, so you answered the question I was going to ask. Thank you.
Of course.
Are there any other questions? Well, if not, let's thank all the students who made excellent presentations this morning, and we will see you all again in the afternoon at 2 o'clock. Thank you. Bye.